I'm just going to show the students what a cloned cauliflower should look like after 3 weeks. Let's see what they think. All right, guys, you ready to see this? This is what hopefully you should have within 3 weeks. So if you gather around, it's worth a look. You ready? Okay, that.
It's got leaves.
What's great about this practical is that it works. And it works because the students use aseptic technique throughout. They begin by wiping down their bench with disinfectant. Their growth medium has a sterilant within it. They soak their instruments in sterilant and they soak their cauliflower florets, their explants, in sterilant as well. That means that the apparatus and the tissue are sterile throughout the investigation, and that prevents the introduction of contaminants. So although it might take several weeks for the students to begin to see growth in their dishes, they won't get contamination by fungus, and at the end of the experiment they'll see what you hope they will see, rather than simply being left with a disappointing dish full of fungus.
We use cauliflower because it's comprised mostly of actively dividing cells and it's readily available. It's also quite a tough vegetable and can withstand some rough handling. The process itself is quite straightforward. You take a floret, break off a smaller bit, and then using a scalpel we're going to cut off a microfloret or an explant. So that's my explant, and it's ready to clone. The next step is to disinfect it, and for that I use a sterilising solution. That's why I'm wearing the gloves, so don't splash any. And it's very simple. I just drop my explant into the solution and give it a good swirl for about 5 seconds. I'm going to leave that there for 15 minutes, giving it another swirl every 2 or 3 minutes. At the same time, I'm going to put these forceps into a beaker of the same sterilising solution. The key to the success of this technique is this stuff. It's an agar growth medium that was developed for use in conservation work. It's already been used to save endangered species. Once the 15 minutes are up, you take the explant and put it into this medium. So that's now ready. Your students can put their name and the date on it and just leave it by a sunny window for about 3 weeks, and after that they'll have their very own cauliflower clone.
It surprised me how precise you've got to be with some of the stuff. How important it is to sterilise things, not to contaminate them, stuff like that, and how that could affect your end result.
It looks really basic, but it's really interesting.
I think it's really interesting, mainly because it's what professionals do.
With this practical, you can bring current techniques from the world of research and conservation into your school. Your students can use the techniques for themselves, and they'll find a real sense of ownership of the cauliflower they've cloned.
The agar jelly was more solid than I'd expected, so I found it a little bit difficult to push in the microfloret. But other than that, it was fine.
What's happened?
I think I'm going to push it in a little bit too deep.
You think you pushed it in a little bit deep? The practical itself doesn't take very long to do, but it's a powerful way to complement the teaching of the theory around cloning. It can be used to demonstrate asexual reproduction and mitotic cell division. It's also a compelling demonstration of totipotency, that is that almost any cell in a plant can be used to produce a new plant.